At this juncture, let me introduce our moderator for today, Dr. Sherry Christian, who will guide us through exploring the role of fat cells in cancer. I had the great pleasure of graduating with Sherry from the University of British Columbia, where she did a PhD in the Department of Microbiology and Immunology. She graduated in 2004 and took a detour postdoc at the University of Alaska Fairbanks. She then went on to Memorial University of Newfoundland for her second postdoc. And Dr. Christian began her position in the Department of Biochemistry at Memorial University in 2010. She is the co-lead of the Atlantic Cancer Consortium under the umbrella of the Terry Fox Research Institute, Marathon of Hope Cancer Centers Network, and the TFRI Atlantic representative. Her lab has multiple research areas with a strong focus on the interactions between fat cells and breast cancer cells, as well as interest in extravesicular biology as it pertains to B lymphocytes.
Well, thank you, Michelle, and thanks for the invitation to moderate this session. So I'm just going to start with, just get right in there, start with a very, very broad overview about fat cells and the tumor microenvironment and why they're really, I find them really, the whole interaction quite interesting. So to start off with, we need to start off with obesity. So obesity is a very quite a big problem in the Western world these days. There's more and more people who are becoming obese. There's a lot of reasons that people become obese. Of course, lack of physical activity and unhealthy diet, but also genetics, mental health, medications, and socioeconomic status. And that leads to fat cell hypertrophy and hyperplasia. So fat cells get bigger, but there's also more of them, chronic inflammation, and other associated symptoms, including an increased risk of cancer, specifically the 13 different types of cancer that I've listed here. And overall, if we take a step back and look at the different types of cancers and the association with obesity, we can see that 4 to 8% of all cancers are directly attributable to obesity. Obesity increases the risk of death by 17% and increases the risk of recurrence by 13%. So being overweight and or obese increases your risk and your chance of death, unfortunately. So why? Well, there's lots of reasons. In some cancers, we have release of estrogen from the white adipose tissue, so the fat tissue itself. In some cases, we have more insulin, and that hyperinsulinemia promotes proliferative signal through the insulin pathway. White adipose tissue is not just a storage place for energy, for fat, but it also is really important, a regulatory organ. So it releases adipokines, which are just cytokines released from adipose tissue. And all of these things can promote proliferation, can induce chronic inflammation, induce DNA damage, and we can get inhibition of anti-tumor immunity. Adipocytes can also sequester chemotherapeutic drugs. And so there's this need to actually adjust the dosage of chemotherapeutics depending on how overweight or obese a person is, and many, many others. And so the problem, of course, is that we don't know, there's no one thing. And so that's why a lot of the research is super important these days to get into why we have these effects. The adipose tissue is really altered by the tumor microenvironment. So this is an example of a number of tissues. And so you can see in the normal tissue, we have these really big adipose cells that are really, basically you see them because they're white empty spaces. And then we have when you get invasive carcinomas, you get movement that you push away that adipose tissue. So physical changes, but also the cancer uses the adipocyte itself. So there's this complex bidirectional interplay between cancer cells and adipocytes. So this is just a little bit of an example. The adipocytes, the fat cells, can release fat, free fatty acids, into the environment that are taken up by the cancer cell. And this allows the cancer cell to use this energy to proliferate. And then the cancer cells can release many different things, many different proteins. And in this illustration, we're showing extracellular vesicles that can then affect the adipocytes. So it's this circular interaction. So we don't have a clinical talk today. And one of the reasons we don't have a clinical talk is because there's not a lot of work going on in the clinic yet. We're still really at the basic research level. So when I was looking for intervention studies, basically I found nothing. There is studies looking at fat cells as treatment. So we can try to turn white adipocytes into brown adipocytes, transplant them next to the cancer, and then those brown adipocytes that are really geared to making energy outcompete the cancer. Or we can take stem cells that are derived from adipose tissue or extracellular vesicles from those stem cells and try to use them as a drug delivery. So take the cells out, load them up with drugs, and put them back in. There's been a little bit of work also looking at diet to affect treatment. So looking at reduced caloric and nutrient intake to reduce disease in ALL. And that has had some effects, but I would say diet hasn't had as many effects as we would have thought. So the reason we don't have a clinical talk today is because there just isn't enough work done yet in the clinic. So with that very quick overview, I'm going to introduce the first speaker, and who's Grant Kelly. And Grant Kelly is actually a PhD student in my lab at Memorial University. And his research is really focused on the discovery of novel molecular interactions between breast cancer and fat cells. So that big question mark at the end, like with the other things. Grant was the recipient of a CGSM award from CIHR and a Cancer Research Training Program award from the Beatrice Hunter Cancer Research Institute, which is a virtual institute in Atlantic Canada that supports cancer research. And today he's going to tell us about some of the work we've done to explore the link between breast cancer metastasis and fat cells in the lab. Okay, take it away, Grant.
Okay, yeah, thank you for that lovely introduction. So as Sherry just mentioned, I'm going to be talking a little bit about the molecular interactions between breast cancer cells and fat cells, their relation to metastasis, and how that all ties into the physical properties of the breast cancer cells themselves. So first things first, just a little more specific data on obesity and how it relates to breast cancer. So breast cancer is the most commonly diagnosed cancer in women and is the second deadliest cancer behind only lung cancer. And it is heavily linked to obesity. So obesity increases breast cancer mortality by as high as 30%. So obese breast cancer patients, they often have larger tumors and are at much higher rates of metastasis, where the cancer spreads to distant other tissues in the body. And the risk changes depending on your hormonal status. So premenopausal obese women with breast cancer are at higher risk for a more aggressive breast cancer, while postmenopausal, they're at a higher risk of a less aggressive breast cancer. So it's very important for premenopausal women to be aware of this and maybe think carefully about managing their weight. If they're specifically at risk for obesity, maybe they're genetic factors. A little bit of background on how we kind of classify breast cancer, some of the properties of breast cancer. So in general, we have a molecular classification system. So there's 4 different groups: triple negative, HER2 positive, and luminal B and A. And these are classified based on receptors present or not present on the cell surface. So we've got 3 receptors. We've got the estrogen receptor, progesterone receptor, and this other receptor called HER2. And so the general kind of rule of thumb is the less of these receptors you have, like in the triple negative, the faster growing, more aggressive and invasive your cancer is. And the more receptors that you're expressing on your surface, the less proliferative, the less invasive and aggressive is, and the more likely you are to actually overcome your breast cancer. So another interesting physical aspect of breast cancer cells is their ability to alter their shape to fit their needs. So I've got here, this is showing, we kind of classify cells shape into two distinct categories, your epithelial or mesenchymal cells. So on the left, you've got epithelial cells. These are immobile. They have lots and lots of cell-cell interactions. They're very sticky. They don't really move that much. And over on the far right, you've got mesenchymal cells. These don't interact with other cells that much. They can freely move around the body a lot easier. So often tumors are mostly epithelial, right? They're these big blobs of sticky cells. They don't express a lot of what's called vimentin. This is a mesenchymal marker, but they do express a lot of ecadherin, an epithelial marker. But under certain conditions, these breast cancer cells can actually shift their physical shape towards this mesenchymal shape, losing this stickiness and gaining the ability to move more freely. So in the middle between epithelial and mesenchymal, there's this hybrid EM state where the cells are both they're both expressing epithelial markers, ecadherin, and mesenchymal markers, vimentin. And this is the kind of metastatic sweet spot, right, where they're highly they're going to be able to move through the body and invade new tissues, but also are still interacting with each other and can maybe form a new tumor somewhere else. So kind of a overview of how this is related to breast cancer and adipocytes. So in the breast tumor environment, you've got adipocytes near the tumor. They're going to release adipokines into the environment. The breast cancer cells are going to react. They're going to undergo what's called the epithelial to mesenchymal transition, right? They're going to lose their stickiness, gain the ability to move through circulation a lot easier, and they're going to move to somewhere like the bone, or the bone marrow, the brain, the lung, the liver, and they might undergo the reverse transition. So they'll lose their mesenchymal properties, go back to being sticky epithelial cells, which is much better for growing a new tumor. And whether or not adipocytes are playing a role in this stage of the process is kind of where my research is mostly focused. So going into a bit more detail about the primary and secondary tumor sites. So a tumor is a really complex network of a bunch of different cell types. You've got your cancer cells, immune cells, macrophages, fibroblasts, and adipocytes, fat cells. They've also got a stiff extracellular matrix, which is the network of proteins that supports cells. So the adipocytes and the breast cancer cells, they kind of bidirectionally talk with each other, and that promotes this epithelial mesenchymal transition. So you form these hybrid EM cells that are both epithelial and mesenchymal. They're really ideal for metastasis. They move throughout the body, invades, maybe bone tissue, brain tissue. But you need to actually establish those hybrid EM cells need to establish a new tumor. So this requires a couple things, but like ECM remodeling. So the ECM, the extracellular matrix at the secondary site might not be the same stiffness that you find in the primary tumor. So that has to change. The breast cancer cells have to regain their epithelial traits. You've got to recruit immune cells and different fibroblasts from the surrounding regions to the tumor. And we don't really fully understand the role of adipocytes in this. So how do we test that? How do we discover that? Well, what we can do is use what's called 3D culture. So normal in the lab, we grow cells in what's called 2D. So this is just on a plastic plate or a plastic plate. And in 3D culture, we actually use extracellular matrix to kind of remake the environment you see in the body. And this is much better at modeling the epithelial to mesenchymal transitions and mesenchymal to epithelial transitions. Does a much better job at giving the cells their natural state. So you can see here, the more epithelial cells are round, and as you progress towards mesenchymal, you progress through the mass-like, the grape-like, and the stellate morphology. So we designed a system by which we could 3D culture breast cancer cells and adipocytes together. And it looks something like this, where we have a layer of adipocytes. We overlay on top of that some extracellular matrix, and then the breast cancer cells go on top. So what happens when we do this is, if I bring your attention over here, we have a no adipocyte control here. These are MDMB 231 cells. This is a type of triple negative breast cancer cell, so the highly aggressive, invasive one. So without adipocytes, we see that they're taking on a very stellate morphology, right? There's lots of branching, the cells are really long, they're kind of not close together. But if we bring adipocytes into the mix, the cells are rounding up. We're seeing lots of small circular colonies. We're not seeing a lot of branching. We're seeing lots of cell-cell interaction. And that's also the same happens if we take conditioned media from adipocytes that we've grown in a different plate and then treat the breast cancer cells with that. So we see a less distinct change, but we do still see some loss of branching, and we have a lot rounder cells. This has been found in a few different cell lines. But what we found is that mostly in aggressive triple negative breast cancer cell lines, we're seeing this change, not so much in our ER, PR positive, HER2 negatives. This would be luminal A cell line. And another thing to note is that because this change is conserved with conditioned media, that tells us that whatever is causing this is being secreted by the adipocytes into the environment and then taken up by the breast cancer cells. It's not a direct interaction. So we looked at some of these breast cancer cells using immunofluorescence. So we basically take antibodies that are designed to stick to a certain molecule that we're interested in, and they have a glowing protein or molecule attached to it, and then we look at it with a microscope. And so when we look at the epithelial marker, ecadherin, or claudin-7, without adipocytes in our MDMB231s or HS578Ts, these are the triple negative breast cancer cells that showed the biggest difference. We see that without adipocytes, we don't really have much of these epithelial markers. When we introduce adipocytes, we see lots and lots of these epithelial markers are being expressed. Right? And the same thing when we just use conditioned media from adipocytes, we also see an increase in ecadherin. Additionally, looking at the proliferation marker Ki67, so this basically tells us, well, how quickly or how the cancer cells are growing. A lot of Ki67 means that you're proliferating quite a bit, and if you reduce Ki67, it means your cancer cells are not growing quite as highly or quite as fast. So regardless of whether or not the breast cancer cells are adipocyte free or with adipocytes or with the conditioned media, we have about the same amount of Ki67. So what this is telling us is that the breast cancer cells, when treated with adipocytes or when they're interacting with adipocytes, they're getting stickier, but they're still growing at the same rate, which is pretty bad, right? So the adipocytes are making them sticky. They're still going to continue to grow. So they're going to form these, big colonies that are pretty ideal for eventually growing into a full-size tumor. But as Sherry mentioned in her introduction, this is not a one-directional interaction. The breast cancer cells themselves are actually affecting the properties of the adipocytes. So what we see is a reduction in the adipocytes is a reduction in the size, but not the number of lipid droplets. So lipid droplets are how the fat cells are storing their fats inside the cell. There's these collections of fats called lipid droplets. And so we see here, these are just mature adipocytes. And this BOAT-EPI 493503, this is just staining the lipid droplets. And so in the control without breast cancer cells, we've got a good amount of really big splotches of lipid droplets. But when you introduce breast cancer cells, you see that the droplets are actually reduced in size. And this is shown in the graph here, where we've got our different breast cancer cell lines, and the number of these droplets aren't really changing. But here, over here, we have different size groups of lipid droplets. The different cell lines are in different colors, and this line here represents the control frequencies of each size. So you can see for the 231s, the 578Ts, which were the most affected by adipocyte co-culture, that the size of these lipid droplets is almost always below the frequency that they are in the control. Right? So on average, we have much smaller lipid droplets, and the amount of larger lipid droplets is greatly reduced. So I mentioned before that because the adipocyte conditioned media also produces this effect, we know that it's a secreted mediator that's causing it. It's not a direct interaction. But we further went on to look at adipocyte conditioned media versus adipocyte conditioned media that was grown with ACM or with extracellular matrix, so this AECM. And we found that if you just have regular adipocyte conditioned media, so no extracellular matrix, you don't actually see any change, right? The breast cancer cells maintain this their stellate morphology. But only when you have ECM do you actually see this change, the losing of the branching and the rounding and bunching up of the cells. So that tells us that whatever is being secreted, it's necessary for the presence of this extracellular matrix. So in order to identify that, what do we do? Well, we took the adipocyte conditioned media and compared to the adipocyte conditioned media with extracellular matrix, and we looked at what proteins were actually present. And so we found that there were 219 proteins that overlapped and 48 that were unique, solely unique to the adipocyte conditioned media with ECM. And then if we looked further at the identity of what these proteins are, and we looked at which ones were the most upregulated or the highest in the AECM, we generated a kind of a top 4 hit list of 4 different proteins that we thought might be contributing to what's going on. So we did some further analysis, and we looked at, okay, well, what are these proteins actually doing? Right? So we wanted to have, oh, my screen just went black there for a second.
Are we still
Sorry about
see your
that.
screen?
No, yeah, okay. Sorry about that.
Oh, it's just, there we go. Yeah.
Is it good?
Yes.
Okay. So we wanted to find a protein that was secreted directly into the extracellular space, but not packaged into a vesicle, which is a little membrane-bound particle that can contain some cargo, and it has a function associated with the extracellular matrix. And so only one protein met that criteria of the top 4, and that was protein lysine 6 oxidase, or LOX. So we wanted to kind of verify this result, and we looked at the mRNA level to see if in adipocytes that we treat with extracellular matrix, does that increase the amount of LOX mRNA? And we found that wasn't the case. But when we looked at the protein concentration in ACM versus AECM, we found that yes, the presence of extracellular matrix caused an increase in LOX protein concentration. So from there, we wanted to see if just LOX protein alone was going to cause this loss of the mesenchymal characteristics. So these are just MDA MB231 triple negative breast cancer cells. They're grown on extracellular matrix. There's no adipocytes. And when we added recombinant LOX, so LOX that we made using bacteria, we actually saw a significant reduction in the number of branches. So these are the number of branches between the cells. So this tells us that while LOX is reducing the mesenchymal characteristics of these cells, it's not alone producing the full effect, the full mesenchymal to epithelial transition. So when we looked at the mesenchymal and epithelial markers, is ecadherin and vimentin, we saw something that kind of where we saw a reduction in vimentin. So here we've got our MDMB231, our breast cancer cells, treated with extracellular matrix. So this left lane is our control, and this right lane here is our added LOX. And so we don't see much in the way of epithelial markers, which is to be expected. These are highly mesenchymal cells, and it's not being increased by the addition of LOX. There is a reduction in vimentin. So this is, again, a partial shift towards the epithelial phenotype, but it's not full. We're not seeing that full effect that you get when you co-culture with adipocytes. And these NMU-MG cells, these are a positive control for the epithelial markers. So that is not really related to what's going on here. And similarly, in this graph, this is just looking at the intensity of vimentin compared to the load and control beta-actin, and we do see that there's a significant decrease when we add LOX. So the future directions and kind of what my work is focusing on is now is inhibiting LOX in the actual 3D co-culture. So we tested recombinant LOX, the addition of LOX protein, to just breast cancer cells, but now we're interested in showing that LOX is a necessary component of this interaction. And to do that, we're going to actually use genetic or chemical inhibitors to prevent LOX activity in 3D culture. And then hopefully the lack of any functional LOX is going to lead to these breast cancer cells that are epithelial normally when they're co-cultured, regaining some of that stellate morphology. So that's currently ongoing. It's part of my PhD work. Another future direction that I'm working on is looking at the role of LOX regulating enzymes. So when LOX is secreted by adipocytes, it's actually inactive, and it needs to be activated by one of two enzymes, BMP1 or ADAMTS2. So these enzymes are going to cut this LOX into two pieces, LOX called active LOX and what's called LOX-PP. So both of these have signaling activity, but LOX is the only one with an actual protein activity and enzyme activity. And that activity is to remodel the extracellular matrix in and around the cell. And so by genetically inhibiting BMP1 or ADAMTS2, we're hoping to determine if one or both of these are involved in this MET process. Okay. And not my work personally, but work that our lab is doing is actually extending this in vitro work to an in vivo metastasis model. And so this is using immune competent mice. We're going to inject them with 4T1 tumor cells. And we're going to, over the course of tumor growth, we're going to feed them with chemical LOX inhibitors and then measure that impact on metastases in the bone marrow. So we're hoping to see that those that are the mice that are fed LOX inhibitors are actually seeing less metastasis to the bone marrow. And then continuing on from that, we're actually going to be looking at doing a tissue-specific LOX knockout. So genetically inhibit LOX specifically in adipose tissue before injecting with tumor cells. And then as the tumors grow and you develop metastases, just feed them a normal diet. And then at the end, measure the bone marrow metastases. And ideally, the mice that are lacking LOX in adipose tissue are going to actually have less metastases to the bone marrow. And that's everything I have. I would just like to say thank you to my supervisory committee, my lab mates, the members of the Villoria PT lab over in OVC at Guelph. And a big thank you to Carol Ann Cole, who has funded me twice now over the course of my research. She's been a major help. If you're interested in learning more about how Carol Ann Cole has contributed to cancer research, I'd recommend checking out the Comfort Heart Initiative over on the Canadian Cancer Society. They sell these lovely pendants that you can, I think it's $20 a pendant, and all of that money goes to funding cancer researchers like myself.
Great. Thank you very much, Grant. It's great. Nice overview. So we're going to move on to the next speaker. And if you have questions, please put them in the chat, and I will get to them at the end. So our next speaker is Professor Catherine Muller-Stomach, who is the head of the Microenvironment, Cancer, and Adipocytes team and the Deputy Director of the Institute of Pharmacology and Structural Biology in Toulouse, France. After training as a clinical hematologist in Toulouse, she actually completed her PhD in multi-drug resistance and with a 2-year internship at the Ontario Cancer Institute in Toronto. So she has a nice connection to Toronto. In Toulouse, she was appointed professor at the Faculty of Pharmaceutical Sciences, and in 2018 created a team focused on adipose tissue, obesity, and cancer. And specifically, her team was the one to identify cancer-associated adipocytes, those adipocytes that are in close proximity to the tumor, demonstrating that these adipocytes contribute to cancer progression, looking at breast and prostate cancer primarily through soluble molecules, extracellular matrix, and lipid mediators. So lots of what we've just heard about. So with that, I will pass it over to Catherine.
So I am ready. So thank you for the invitation. As you just say, I have a big connection with Canada, and I must highlight that my own daughter is living in Canada now. So, it's almost my second country. So I will show you our result about fat cell filling cancer cell and try to highlight some potential targets. So I will just go quickly over that because it has been already shown. We start with the fact that obesity affects cancer progression, and it worsens the prognosis of mainly ER breast cancer, independently of menopausal status, but also prostate and colon cancer. And at diagnosis, you have tumor of higher size with increased local and distant dissemination. And over the course of the cancer, you have increased distant dissemination and resistance to treatments. I just show you big studies that have been published quite so long ago, showing that the distant metastases increased by almost 50% in obese patients. So as it has been already highlighted, there is several explanations for that. Especially delay in diagnosis. Maybe the patient with obesity are less prone to participate to screening campaign. There is also, but it was highlighted, the fact that there is inappropriate therapy, especially inadequate dose of chemotherapy. But the hypothesis that we develop is the fact that we may have aggressive tumor related to the host, and we wanted to study the role of adipose tissue. So I just want to highlight some fact about adipose tissue. The first one is that we have multiple adipose depots throughout the body. There are subcutaneous adipose tissue and also visceral adipose tissue that has been well described. But there is also what we call organ-associated adipose tissue, such as mammary adipose tissue, periprostatic adipose tissue, or bone marrow adipose tissue. And there is more and more evidence that these adipose depots exhibit specificities. So adipose depots are composed of adipocytes and other cells that we call stroma vascular fraction. And these cells are embedded in extracellular matrix. Adipose tissue has two main cell functions. The first one, and I really want to highlight it, is a metabolic function, meaning that the fat cells are able to store fatty acid and to release them upon energy deprivation or effort to deliver fatty acid to liver, heart, and muscle. And adipose tissue, it's also endocrine and paracrine tissue, and they are able to secrete what we call adipokine, and today there is more than 400 adipokine identified. In obesity, you don't you don't only accumulate more adipose tissue, but the adipose tissue is dysfunctional. You have hypertrophic adipocytes that will die if they are becoming too big, and it leads to the recruitment of inflammatory cells. And it is now admitted that in obesity condition, the adipose tissue exhibits sub-inflammatory state and fibrosis, but this is mainly described for subcutaneous and visceral adipose tissue. So really what we work on in the lab is what happened to the adipose tissue near cancer. It's what we call paracrine hypothesis. It has really been shown that obesity also affects circulating factor, but really we work on what's happened to the adipose tissue close to the tumors. With the hypothesis that the proximity of obese adipose tissue with cellular and metabolic reprogramming could affect the tumor cell behavior. But when we start the project, we don't even know if adipocytes participate in tumor progression. So it was really the first question we asked. So to answer to this question, we make, I would say, simple experiments by co-cultivating adipocytes with breast cancer cell, and we observe the change in adipocyte. So as you see here, we found that there was a delipidation in adipocyte. It's a red oil labeling. We also observed that there is a decrease in late adipose markers, meaning that you have a dedifferentiation of adipocytes and what we call an activated phenotype marked by the overexpression of some ECM-related molecule, but also pro-inflammatory cytokine. So at this time, it was very important to look if this exists in human tumors. So as you can see here, at the invasive front of human tumors, we observe a decrease in size of adipocyte. We were able to verify by immunohistochemistry that these adipocytes exhibit dedifferentiation by comparing tumorectomy with reduction mammoplasty, and they also exhibit activated phenotype. And these results were also confirmed. Sorry, my mouse is very sensitive. We also confirmed these results with isolated adipocyte and performing qPCR. So this paper has been published in 2011, and we were happy to see that it has been highlighted as a key landmark in cancer in a recent issue of Cancer Research. So I don't have time to show you this result, but when we incubate the cancer cell with adipocyte for a longer time, we obtain really completely dedifferentiated adipocyte that we call adipocyte-derived fibroblast. So this phenotype, we call this adipocyte cancer-associated adipocyte. It is absolutely not restricted to breast cancer, and it has been shown by a huge amount of team now that as soon as a solid tumor is close to an adipose depot, this process occurs. So it's how you can see what's happening in the tumors. The tumor is able to modify adipocyte into cancer-associated adipocyte, and then in the adipocyte-derived fibroblast. And this fibroblast has increased migratory and evasive ability, and they are able to penetrate within the tumor. So part of the cancer-associated fibroblast probably derived from adipocyte. So all over these years, we have been studying the role of this cancer-associated adipocyte in cancer progression. So there is a lot of molecules that have been involved, including cytokine, ECM molecule, especially by the group of Philip Scherer in Dallas. But I will insist on free fatty acid, because you may know that there is increasing evidence in the literature that tumor cells really are able to take up lipids from outside. And if you consider a tumor, the most important reservoir of lipids close to a tumor is the adipose tissue. The first question we ask is, what are the mechanisms that contribute to the occurrence of cancer-associated adipocyte? As I show you, there is a dedifferentiation or transdifferentiation. I couldn't comment on that if you ask a question about that, but the first question is that, do tumors are able to induce lipolysis in adipocyte? I remind you that lipolysis is the ability of the adipocyte to break down triglyceride and to remove free fatty acid in energy-demanding condition. The end product of lipolysis is glycerol, and we were able to show that when we incubate adipocyte with a tumor-conditional medium, we have an increase in glycerol, showing that they were able to induce lipolysis in adipocyte. So for the moment, the lipolytic signal induced by cancer cell remain a mystery. Some work has suggested the implication of catecholamine, pro-inflammatory cytokine, adrenomedulin, hypothesis that we tested in the lab, and all the results we obtained were negative, so the lipolytic signal is not really well characterized. So the lipids are released as free fatty acid, but also contained within vesicle. So the second question that we can ask, are these lipid transfer in cancer cells? So you can see here that when you co-cultivate cancer cell with adipocyte, you can see accumulation of lipid droplets that you can see here by immunofluorescence or red dot staining. But I'm really sorry. I must point out that it is very important to understand that when you look at that, you look at the ability of tumor cell to re-esterify free fatty acid in triglyceride. So you have two movements of free fatty acid, the free fatty acid that are used very rapidly by tumor cell, and the excess of free fatty acid that are stored in lipid droplets to protect cancer cell from lipotoxicity. So we demonstrate the direct transfer using radiolabel adipocyte, but now it is completely admitted that it is a transfer of lipid from adipocyte. So what are these adipocyte, these free fatty acid, used for? They are used for fatty acid beta oxidation, which is successive breakdown of fatty acid to generate acetyl-CoA. So as you can see here on that slide, you have an increase in fatty acid activity in tumor cells. And when you inhibit fatty acid beta oxidation by etomoxir, which is an irreversible inhibitor of a protein which is called CPT1, which is able to transfer the lipid within the mitochondria, you can see that you increase the lipid droplets, meaning that you have some free fatty acids that are used. And if you block the use of free fatty acid, you increase the storage of lipids within lipid droplets. So when we look at what's happened in terms of cancer aggressiveness, I forgot to tell you that one of the main effects of adipocyte is to stimulate migratory and invasive ability of the cells. We were able to show that inhibiting fatty acid beta oxidation by etomoxir block the cancer invasiveness induced by adipocyte. It also blocks the lung metastasis in mice. And you already heard about that. In the presence of adipocyte, we observed an epithelial to mesenchymal transition in ER-positive breast cancer. And you can see that using etomoxir partially reverse the EMT process. So if we want to sum up, it's a little bit complicated. I would like to say that first, the transfer of free fatty acid from adipocyte into cancer cell has been demonstrated in a wide range of models, but the fate of fatty acid is dependent of the tumor type. In our hand, in breast cancer and colon cancer, the fatty acid beta oxidation was not coupled to ATP production. Whether in another group, it has been shown in melanoma and ovarian cancer that it is linked to ATP production. And in another model, which is prostate cancer, the fate of free fatty acid was to increase ROS production and generate a cascade which stimulate tumor invasion. So the question is that, is this lipid transfer increase in obesity condition? So we go back to the mammary adipose tissue because there was very few data about how obesity affects mammary adipose tissue. So in collaboration with a group in Oslo, we demonstrate that in fact, we have an increase in size of adipocytes, and we have also, what we call crown-like structure. It's macrophage surrounding dying or dead adipocyte. But as you can see, this number of structures were very low in mammary adipose tissue. We also demonstrated that this hypertrophy of adipocyte is correlated to visceral adiposity. And I still don't have time to show you the result, but our latest results show that in mammary adipose tissue, contrary to visceral or subcutaneous adipose tissue, we didn't find any fibro-inflammatory response, meaning that one of the key events in mammary adipose tissue upon obesity is the fact that the adipocytes were hypertrophic. So to answer to this question, whether or not the metabolic symbiosis is increased in obesity condition, we decide to set up a model of culture of mammary adipocyte because all the results I showed you before were performed with adipocyte that has been differentiated in vitro. They are obtained from pre-adipocyte cell line. And as I told you, there is an increasing amount of data in the literature showing that there is differences between adipose depot. So we set up a model where we cultivate the adipocyte in a fibrin matrix. The interest of fibrin matrix is the fact that it's polymerized very quickly. So you have all these adipocytes that are distributed homogeneously. And this matrix is not digested by tumor cells. And as you can see here, we were able to cultivate this adipocyte for 5 days, and they remain at the same size. And we have this difference between obese and non-obese adipocyte. And we also show that these adipocytes are still functional, meaning that they are able to respond to lipolytic signal. Using this model, we were able to demonstrate that there is a transfer of lipids which is increased in obesity. We co-cultivated tumor cells with adipocyte, and you can see that there is lipid droplets within a tumor cell, and the number of lipid droplets and the size is increased under obesity condition. So for the moment, we are investigating what is the effect on tumor cell of this increased lipid droplet. And there is another question, which is very important, is the fact that the lipid transfer has never been demonstrated in human tumors. There have been many obtained in vitro co-culture systems, so we really don't know if this lipid transfer exists in human tumor. So to answer to this question, we are now developing a strategy which is the coupling of mass spectrometry on the same slide with immunofluorescence. The idea, mass spectrometry allows you to identify lipid metabolite within the tumors, and by using immunofluorescence, we are able to identify in which cell which metabolites accumulate. So I show you very preliminary results where we have been labeling tumor cell with cytokeratin, and we investigate the lipid content in tumor cells that are distant from adipocyte or within tumor cells that are close to adipocyte. And our preliminary results suggest that we have an increased triglyceride accumulation in tumor cells that are close to adipocyte in human breast tumor. So we are really working on that aspect right now. So to sum up and to discuss a little bit of what happens, we know that adipocytes are able to release free fatty acid. The signal that contribute to this release of free fatty acid is not known. You have several key targets, which could be the transporter, because there is a need to transport the free fatty acid within the tumor cells. Once free fatty acid are in the tumor cells, there are a group of proteins which are called fatty acid binding protein that are able to redirect the free fatty acid to several compartments of the tumor. They are able to contribute to the storage and resterification in lipid droplets. They also contribute to send the free fatty acid to the membrane and to the nucleus, and also to direct them to mitochondria. So the metabolism and the fatty acid beta oxidation activity is another target. And I didn't show you the results, but when the tumor cells store free fatty acid in lipid droplet, they are able to release this free fatty acid over time. And this lipolysis activity may be also a target. What we can say for the moment is that the membrane lipid transporter are really, the membrane lipid transporter are really cancer type dependent. Most of the work that are currently ongoing in on CD36 that has been shown to have an important role in prostate cancer and lung cancer. In our end, in breast cancer, we do not have CD36 overexpression, but there is a blocking antibody that works well in a preclinical model in the several types of cancer. Among FABP family, FABP5, which is overexpressed in a lot of cancer, is an interesting target, but there is only inhibitors that can be used, in vitro. You can speculate to block metabolism by using FRO inhibitors. So etomoxia is not okay because it's irreversible inhibitors, but there is other inhibitors that have been developed and enzyme involved in fatty acid beta oxidation, and they are currently under clinical trial in diabetes and cardiovascular disease. So we may expect that if the safety is okay, it can be envisioned in cancer. The mechanism of lipolysis in cancer need to be better described. And as I told you, the inhibition of tumor-derived lipolytic signal is a very good idea, but the signal is still unknown. I just, to finish, must to point out that the result has been mainly obtained in vitro and in preclinical model. And for me, there is really a need of large clinical study in lean and patient with obesity to validate the target and to see the difference between the two population. So just to finish, my team in Toulouse, I just would like to thank all our collaborators and especially the surgeon and pathologist, because you know when you are doing that sort of research, they are mandatory, and all the foundation that support our work. So thank you very much for your attention.
Thank you so much, Catherine. Would you be able to, I'm going to invite each of the speakers to give a one take-home message, and then we'll move on to the Q&A. So, Catherine, since you're up still, is there one take-home message that you think people should take from your work or from this field in general?
I think obesity is a very important key factor in cancer progression. And I think that the lipid content of adipocyte is probably key to this effect.
Thanks. Grant?
Yeah, I think we really just know so very little about the kind of breadth of interactions between adipocytes and breast cancer cells. And there's just so much to look into. There's so many different things that we can explore, and really just it to me is like a really big part of kind of the future of our understanding of how cancer really works within the body.
Mm-hmm. Thanks. And I think I want to reflect a little bit on something that Catherine was saying about the different depots. We don't know enough about the different depots. And I think that's going to be a really important area to work in. Mammary and subcutaneous and visceral but bone marrow, they're all probably quite different.